Hello, friends. Welcome to Science Talk. I am your host, Jim Massa. Okay, this is an extremely lengthy article. It's actually a literature review, but I want to discuss with you what we see right here: plastics and the microbiome impact and solutions. This was published early part of this year, and it was published in Environmental Microbiome. I will leave the URL to this link here in the comments section below. It's very lengthy, so I'm not going to cover every aspect of it because I will probably be yammering away for about 3.5, 4 hours. But you can then click on the URL and read it in detail at your leisure and discretion. So let's at least look through the abstract here. Global plastic production has increased exponentially since manufacturing commenced in the 1950s, including polymer types infused with diverse additives and fillers. Excuse me. While the negative impacts of plastics are widely reported, particularly on marine vertebrates, impacts on microbial life remain poorly understood. Plastics impact microbiomes directly, exerting toxic effects, providing supplemental carbon sources, and acting as rafts for microbial colonization and dispersal. Indirect consequences include increased environmental shading, altered compositions of host communities, and disruption of host organism or community health, hormone balances, and immune responses. Okay, let me pause there for a moment, kind of give a little summation here. Basically, what they're saying is that the microbiome, there could be, think, for example, the microbiomes that's located in your digestive tract, your guts. It will impact that. I did a video where I talked about nanoplastics in the food. We're ingesting the plastics, and the plastics disrupt metabolic processes, basically messing up the thyroid gland, and such that the result is, especially if someone has a genetic predisposition for being adversely affected, the result is the person becomes obese because the hormonal releases are all affected. There's an imbalance, and it can lead to hypothyroidism, which will then lead to weight gain. I'm an example of that. So we have that. I also did another video some time ago about where I discuss larval fish ingesting plastics, and included a video of this happening. Here's the larval fish, and there it is ingesting plastic bits here. Okay, so basically, there are other health issues that are going on that have been demonstrated with ingesting the plastics, including cardiovascular diseases, cancers, uh, problems with the immune system being negatively impacted. And plastics are everywhere. Take the oceans, for example. They're present in the Arctic Ocean all the way down to the Mariana Trench. They are everywhere. And we basically have ingested plastic. There's some estimates that we can ingest up to basically a credit card size amount of plastic per week, or at least per month. But we are ingesting plastic. You'll probably find it in our fecal matter. So the isolation and application of plastic degrading microbes are of substantial interest, yet little evidence supports the microbial biodegradation of most high molecular weight synthetic polymers. High molecular weight, very long chain chemicals. So the molecular weight is high. Synthetic polymers typically are difficult to be broken down, and typically they are not. Over 400 microbial species have been presumptively identified as capable of plastic degradation, but evidence for the degradation of highly prevalent polymers, including polypropylene, nylon, polystyrene, polyvinyl chloride, PVC, must be treated with caution. Most studies fail to differentiate losses caused by the leaching or degradation of polymer monomers, additives, or fillers. Even where polymer degradation is demonstrated, such as for polyethylene terephthalate, the ability of microorganisms to degrade more highly crystalline forms of the polymer used in commercial plastic appears limited. Microbiomes frequently work in conjunction with abiotic factors such as heat and light to impact the structural integrity of polymers and accessibility to enzymatic attack. Consequently, there remains much scope for extremophile microbiomes. These are basically bacteria that can live in extreme harsh conditions. And microbiome, of course, would be like a community of them, to be explored as a source of plastic degrading enzymes and microorganisms. We propose a best practice workflow for isolating and reporting plastic degrading taxa from diverse environmental microbiomes, which should include multiple lines of evidence supporting changes in polymer structure, mass loss, detection of presumed degradation products, along with a confirmation of microbial strains and enzymes along the associated genes responsible for high molecular weight plastic polymer degradation. Such approaches are necessary for enzymatic degraders of high molecular weight plastic polymer to be differentiated from organisms only capable of degrading the more labile carbon, while predominantly amorphous plastics, plastic monomers, additives, or fillers. When you look at, take for example, the garbage patch there, a lot of that plastic there is this synthetic material that is very difficult to break down, and it accumulates. And you have the garbage patch, for example, in the Pacific Ocean, the North Gyra, about the size of France's Texas, or something like that. It's it's a major problem. I saw a recent video of, I forget where, I think it was in Thailand, someplace like that, where it's just literally a river of plastic flowing down the street. I mean, it was just insane. The first plastic to be produced in commercial quantities, Bakelite, was invented in the early 1900s. A scarcity of resources and a need to enhance technologies following the First World War drove the development of new and improved synthetic materials, including plastics. Plastics now constitute a large, diverse group of materials made from combinations of synthetic and semi-synthetic polymer materials, frequently incorporating additives which aid the manufacturing performance of the final product, such as plasticizers, antioxidants, flame retardants. Now, here's the important bit here. I'm sure you're all aware of this, but we need to emphasize this. Plastics are predominantly derived from fossil fuels, for example, oil, natural gas, although they may also be made from renewable resources such as quote unquote, bio-based plastics derived from cornstarch or sugar beet. Plastics such as polyethylene terephthalate, PET, may be synthesized from either source and sometimes referred to as drop-in plastics. With the onset of mass consumerism in the 1960s and a move away from the use of traditional natural materials to more versatile plastics, plastic is now an integral part of our everyday lives. Plastic production has increased exponentially since the 1950s, with an estimated 8,300 million metric tons of virgin plastic being produced to date and an expected annual production rate of 1,100 tons by 2050. They go on to describe, other types of, fossil fuel-based biodegradable polymers such as polycaprolactone, PCl, polybutylene adipate terephthalate, PBAT, currently not used in large scale. Less than 1% of polymers are bio-based. less than 1%. That means the rest are oil-based. And of this less than 1%, 44.5% are drop-in. So they share similar properties to the varieties made from fossil fuels. They are considered non-degradable. Of the almost 300, let's highlight this is important. Of the almost 360 million tons of plastic produced annually, only a small fraction, about 1%, is bio-based. So what they're saying there is plastics are being derived from fossil fuels. So at the end of the life, essentially three fates of plastics: recycling, incineration, and discarding. All right? And life management plastic product has not kept pace with rapid increases in production, so it's getting away from us. Only 10% globally are estimated to be recycled, 14% incinerated, the remaining 76% goes to landfills or enters the natural environment. For example, the oceans, the rivers, in the ground, right? 76% ends up in the environment. Organisms can ingest this. I've seen reports of cetaceans washing up dead, and when a necropsy is performed, their stomach was packed full of plastics. Had a full stomach, but the animal was starving to death. And of course, we know about turtles and pinnipeds getting tied up and wrapped up in the plastic, or that they drown, or they can't swallow food because their throat is constricted. We've all seen that. Okay, so microbial impacts of global plastic pollution. As I said here, I'm not going to go through every single word here because we'll take a look at it. I'm only down to here, right? And the thing goes all the way down to there, so it will take several hours. I'm hitting the highlights. I will post the URL link in the comments section for those who want to read this in more detail. But I just want to kind of bring a synopsis of the latest science in this, what the situation is, and potential ways to mitigate. The recent death of a Cuvier's beaked whale in the Philippines with 40 kilograms of plastic waste in its stomach, the necropsy of young sperm whale on a Scottish beach yielding 100 kilograms of refuse, well, that caught people's attention. And this is an example of what plastics will do to organisms. More than 800 animal species are already shown to have been affected by plastic pollution, and with an increasing number from the detritivorous sea snails to apex marine predators being found to have internalized plastics, meaning they've ingested it. Globally, Wilcox et al. Predict that as many as 90% of all seabirds ingest plastics. I did a two-part video segment. In part one, I showed a dead albatross, a photo of a dead albatross with plastic all around it. It had decomposed for a little bit, and all the plastic came out of its gut. And then part two was the larval fish ingesting plastics. Postmortem images of plastic spilling from the guts of dissected marine animals are causing us to reconsider unsustainable plastic use. Yet the impacts of plastic pollution on most smaller organisms remain less well studied. Certainly, negative consequences of plastics have been reported for myofauna such as Daphnia magna and Cenohabditis elegans, nematodes. Daphnia magna are freshwater crustaceans. It's you have like Daphnia pulex. There's a whole bunch of species of the Daphnia order. As I said, they're crustacean. They're sometimes referred to as water fleas, and they're widely used in limnological, limnetic studies, lotic, lenic, and all that kind of stuff there for various things such as measuring filtration rate and so forth. Um, but they're finding plastics in these creatures here, and a Daphnia is typically 1 maybe 2 millimeters in length. So this is give you an idea of the size of them. Okay. So and then these nematodes, of course, worms, largely attributed toxicological impact or blockage of the digestive system and related reductions in feeding rates. In contrast, the impact of plastics on environmental communities of microorganisms is rather less well researched. So they're going to define what they mean by microbiome. So let's look at that. The term microbiome describes the combined genetic material or community of microorganisms inhabiting a particular environment. So there's your definition. Researchers will, continue to examine various microbiomes: soil, marine, freshwater, atmosphere, subsurface environment, the flora in an organism's say digestive tract is another example of microbiome. And here it is. The term microbiome is perhaps predominantly used to describe research into the microbiome of the GI, gastrointestinal tract, gut microbiome. And since environmental plastics concentrate in the digestive tracts, they have the potential to impact the gut microbiome. And they say, however, due to the widespread environmental distribution, impacts of plastic pollution further extend to the microbiomes of diverse non-host associated environments. And then they call that the environmental microbiome to distinguish it from the gut microbiome. Direct impact of plastics on gut environmental microbiomes are multiple. Some plastics and/or their associated additives provide organic carbon sources metabolizable by certain microorganisms. However, the microbial degradation of most plastics is restricted to only a few taxa. It is slow process, and there's still some debate about it, if they are effective or not. So there's very little evidence. There's a lack of evidence for microbial degradation of dominant plastic polymers, including polypropylene, polystyrene, polyethylene, nylon, and the PVCs, polyvinyl chloride. Okay, so now let's look at figure 1 here. Let's see if I can pull up a full size figure here for you. There we go. Okay, so perturbations directly. Plastic used as a microbial carbon source. Toxic impact of the plastic or the additive, and plastic as a vector for microbial dispersal. Okay, so let's look at the impact from the direct perturbation. Dysbiosis, so you have increased abundance of pathogen invasive taxa. The community is altered, composition, diversity, and functioning is affected. Indirect, gut microbiome, mechanical, reduced host feeding fitness due to gastrointestinal blockage, which means if there's interfering, you're not going to ingest as much food. And if you do ingest food, probably interference, so you're not going to get the nutrients required out of it. And so basically, the body goes into starvation mode. And enhance immune and inflammatory responses. And then disruption of host hormone balance. I mentioned these at the outset. Environmental microbiome, mechanical, enhanced shading, increased soil water content, biological, altered compositions of higher plant, animal communities, chemical, toxic leachates generated from physiochemical plastic weathering. Okay, so the impacts, we see the exact same impacts for the indirect as we did for the direct. What are some of the potential solutions? Bioprospecting for degrading taxon communities, genes, and enzymes. Enhancing degradation, environmental manipulation, for example, altering the temperature, altering the pH. Biotic manipulation, enhance co-metabolism. In other words, find organisms that ingest, digest, break down, would deal with these materials. And then, of course, genetic manipulation. So, so this is kind of a little summary showing some of the direct and indirect impacts and how this can alter, an organism's behavior or how a community responds as a whole. And usually, the physiological impacts tend to be deleterious. And let's say right here. In this review, we highlight recent knowledge on the direct and indirect impacts of plastics on the health and functioning of environmental microbes, and then as well as gut microbiomes. Then they consider how we can mitigate plastics, etc. Impacts of plastics on the gut microbiome. I'll just highlight a few of these. Plastics cause a variety of undesirable mechanical, chemical, biological impacts on the species that ingest them, or the individual. Right? The consumption of plastics, either directly or via trophic level transfer, I'll have a few words to say that in a moment, has multiple direct consequences in reducing appetite, impacting feeding activity, decreasing body weight, fitness, and fecundity. Fecundity is how many gametes are produced. Now, it's interesting that it said decreasing body weight. There are studies that show that it messes with the the endocrine system, and thus affects metabolism, so that the metabolism slows down and the individual actually gains weight. So it's kind of some interesting results. It can go decreasing body weight or increasing body weight. In severe cases, the accumulation of large plastic masses may block the gastrointestinal tract, and that can lead to death. Smaller fractions of plastic may also bioaccumulate in the body using the gut, but it can also move to the liver and the kidneys. So that implies that the microplastics can go across the gut epithelial lining and enter the circulatory system, where it can then be deposited in various organisms. Now, this bit here about the trophic level transfer. In my nanoplastics video, I discuss how the plastics end up in the plants, and I showed you photos in that video of nanoplastic sized particles inside the xylem and the phloem, inside plant tissues. We then eat that plant tissue, and we now have ingested that material. That's an example of a trophic level transfer. While the physical presence of plastics demonstrably impacts the microbiome gut immune axis, additives which leach from plastic polymers have further consequences. Plasticizers are the largest group of plastic additives, particularly phthalates, which may concentrate in bodily tissue to induce multiple adverse effects. For example, diethyl hexylphthalate, DEHP, causes anti-estrogenic properties in fish, hindering the development of reproductive organs, presumably due to competition with endogenous estrogens for the receptor. Dibutyl phthalates delay gonad development and functioning in mammals and amphibians. And they list through a whole bunch of other examples. Impacts of plastic additives on the gut microbiome remains little explored, although Adamovsky assessed the consequences of environmentally relevant concentration of the widely used plasticizer DEHP on zebrafish. DEHP caused dysbiosis of the gut microbiota. An assessment of the GI transcriptome revealed the upregulation of T cells thought to play key roles in pathogen neutralization by maintaining the integrity of the intestinal epithelia, while downregulating neuropeptide Y, a hormone which can modify immune activity by regulating T cell function. Okay, it's getting a bit wonky. Microplastics are potential vectors of pests and pathogens around the globe via ocean currents. So too may they vector pathogens into the gut. I think they need a word. So too may be they are vectors pathogens into the gut. It's kind of an awkward wording there. What they're trying to say is that microplastics uh, can provide a ride for pathogens and other disease organisms, and then they're transported around through various physical mechanisms such as ocean currents or through ingestions into the gut. That's what they're saying. Microbial attachment of plastic particles can enhance both microbial dispersal and survival, for example, biofilms. Pathogens such as Vibrio parahemolysis causes septicemia, basically nasty infection of the gut, gastroenteritis in humans, and they've been identified in marine plastic and associated biofilm communities. Okay. Impacts of plastics on the environmental microbiome. In terrestrial environments, the mere presence of plastics exerts physical impacts directly impacting microbial communities. Agricultural plastic mulch film applied to enhance short term crop productivity cover perhaps 20 million hectares of farmland worldwide and a significant source of terrestrial plastic contamination. This here was the basis for that nanoplastics video that I did a couple of months back. While research is focused on the impact of synthetic plastic film, the microbial consumption of biodegradable plastics is noted to have profound impacts on soil microbial communities. Once embedded in the soil, plastics impact soil water interaction by increasing water content, a major determinant of soil microbial community composition and functioning. By altering the availability of water, the physical impact of plastics on the soil environmental microbiome may be substantial. The consequence of other physical impacts, such as increased shading by plastics, has been hypothesized to reduce aquatic photosynthesis. Though that's been hypothesized, there appears to be a lack of evidence supporting such a hypothesis. Presence of plastic has direct chemical consequences for environmental microbial communities. Readily biodegradable plastics such as polylactic acid contribute available carbon, in some cases significantly increase microbial biomass enzymatic activity. Presence of such plastic in soils alter community composition, enriching the abundance and activity of certain taxa. Through the ubiquitous interaction between microorganisms and macroscopic plants and animals, plastics and associated compounds exert multiple indirect biological impacts on environmental microbiomes. For example, plants can be impacted as they take up plastics such as polystyrene via their roots, altering the root length, the weight, and oxidative stress responses caused by the disruption of cell wall pores, cell to cell connections used for nutrient transport. Plant taxonomy and health play an important role in shaping soil and rhizosphere microbiome, impacting the quantity and quality of root exudates and the potential of plants to recruit specific members of the soil microbiome and promote the expression of genes. Plastics can also influence the rates and extent of microbial dispersal among environments. Buoyant plastics such as polyethylene, polypropylene, polystyrene are transported over long distance by winds and ocean currents, whereas non-buoyant plastics such as PET, PLA may act as a vector to transport surface-associated microbes to deeper water. This is a mechanism for why we find plastics in the Marianas Trench. Microbial groups, including toxic microalgae, potential human and animal pathogens, have been detected associated with marine and freshwater plastics, along with diverse antibiotic resistant taxa. Plastics are further postulated to vector pathogens through wastewater treatment plants, pest species via ballast water. Ballast water is typically what's in the ships to maintain ballast. And usually, it's a breeding ground for all sorts of microbial stuff. And every so often you purge out the ballast water. Well, now you just dumped all of that into the aquatic system. So, yeah, might want to rethink how we do that particular practice. Okay, assessing diverse plastisphere communities via amplicon and metagenome DNA sequencing. So the development of molecular methods, including high throughput DNA sequencing technology, is increasing our knowledge of the diverse nature of plastic-associated microbiomes. And they go on to say, they list a bunch of different phyla, cyanobacteria, and so on, where they would be found, etc. The specificity of plastisphere communities has been investigated in comparison to communities growing on inert surfaces, such as glass, ceramic, and the results vary. A study by Ober-Beckman and others using 16S RNA gene amplicon sequencing for taxonomic analysis found no significant difference between the pelagic is open water, microbial communities associated with PET, plastic bottles, and glass microscope slides as a control, deployed for 5 to 6 weeks. And then, so they talk about some other studies here, and they're leading the researchers to assume that there may be no specific plastic associated communities. As you can see, is that they report a lot of studies here. You can read through that, form your own assessments on what they're stating here. Considering the longevity of plastic debris in the environment, the relatively short length of most lab-based studies may not be enough to explore the full degradative potential of the plastisphere microbiome. That is an extremely legitimate point, because if we know that plastics have a longevity, they persist in the environment. So you need the time to see what any such processes that these plastics are subjected to, what the effects might be. Do they break down or not, as an example? And if you're doing short base, lab time, elapsed time, you may not really be able to, with justification, extrapolate your results further out into the future. So, some of the questions that remain. Which organisms act as pioneer species when plastic is first introduced into the environment? And do the priority effects of early colonization affect the overall composition and metabolic potential of the microbial community later on? It's very important questions that they are raising there. 2, does there exist a core global community of plastic degrading taxa, or do they exhibit substantial geographic or habitat-specific biogeography? In other words, is this going to vary from region to region, or is there a global community that could perhaps be deployed to mitigate the effects of plastic, break it down, so on? If it's going to be region to region, that presents greater challenges. 3, if core members of the plastisphere vary in abundance between plastic types and biofilm maturity, can the presence and abundance of certain microorganisms indicate the approximate type and age of plastic debris? Another very good question. In other words, okay, do we need to use specific taxa to attack these things? And they say answer to these questions will assist our ability to identify plastic-specific microorganisms from different regimes, biomes on different plastics, different stages of plastic aging and degradation. Additionally, such knowledge likely increases our ability to use microbial community DNA to inform on the environmental impact of plastics. Many been through taxonomic analyses, chemical analyses. They list a whole bunch of examples here. So this will bring us to the end of part 1 on this video series discussing plastics in the microbiome. Part 2 will begin with the section that you see here, mitigation. So stay tuned. Thank you for your time. Hello folks, this is Jim here with Science Talk, asking you to please subscribe to my channel and to inform others of my channel and of the work that I do. Please share to social media platforms that you use. Also, as a reminder, don't forget to click the bell so that you know when I load up more videos. Finally, I ask that you support the work that I do by becoming a patron at patreon.com. Details in the description box below. Thank you for your support.